Okay, so this was a case with what happens when you have you have the alleles which are homozygous. Okay, now we'll see the other condition. What if the alleles are heterozygous? So if the alleles are heterozygous, so this will be the case. Alleles are heterozygous means I am taking it as capital B and small b. So this is the case. So one is capital B, the other one is small b. So there is subtle differences here. What occurs here? So in meiosis 1, they separate capital B in one and small b in the other. So this is solely capital B, this is solely small b. As a result, so this was meiosis 1, reduction division. Now you have meiosis 2, which is same as that of mitosis. So here this is the spindle attachment, here this is the spindle attachment. So what happens here is, here you get this chromatids, sister chromatids get separated. Here also sister chromatids are getting separated, but the difference is these two are capital B's and these two are small b's. So if you are having, or if you are considering a character which is heterozygous, what you can see here is, you can see two different types of gametes. That means one type of gamete with capital B and the other type of gamete with small b. That means either one type of egg with capital B or sperm with capital B gene and the other set of sperm with small b gene. So if you are considering such ones, you will have to, anyway there are 4, but these two are same and the other two are same. In such cases, when you do, or when you write genetic problem, if you are having an individual with capital B small b, you should write its gametes, only 2 gametes, as one capital B and the other small b. If it is homozygous, you get only one type of gamete from this, which is capital B. So this particular rule should be kept in mind. So this is the basic of doing genetic problems. So we discussed what is our homozygous and heterozygous, etc. That's why we discussed this one. So if you are considering a trait or a character which is homozygous, you get only one type of gamete. If it is homozygous, only one type. If it is heterozygous, you get 2 types of gametes. Even though after meiosis you have 4 different or 4 gametes which are being formed, because they are same. If it is homozygous, all the 4 will be same. If it is heterozygous, 2 will be of one type and the other 2 will be of one type. That's why we take 2 gametes here and only one gamete. This particular thing, I mark it again. This should be kept in mind. This is very important in doing genetic problems. Okay, so now we'll discuss our oogenesis and spermatogenesis, and we'll wind up the session. So this particular meiosis is happening in both spermatogenesis as well as in oogenesis, but there are some differences in the sense that spermatogenesis is a process that occurs in males, and that happens inside the testis in structures which are called as seminiferous tubules. And this particular event called as spermatogenesis in males, the case I am talking about, the case with humans. In humans, this particular event of spermatogenesis is initiated in males at puberty, only at puberty. That means this particular meiosis starts in the testis at puberty, and that is a never-ending process. Never-ending in the sense that till his death, this particular process happens. This will happen, but the rate of spermatogenesis will decrease as age occurs, and also other disease condition may also occur, also affect the process. But if he is healthy till death, what is to be hypothesized is the process of spermatogenesis is never ending after when it starts with puberty and till that the process occurs. So that's with spermatogenesis. Coming to oogenesis, this is a process in female, or that happens inside the ovary, and this process is initiated in a female even before her birth. That means even the process starts in the womb of her mother. Okay, this particular process, when you compare it with spermatogenesis, as I said, it is a never ending process. This particular process is having a halt in the lifetime of a woman. Okay, and the halt is nothing but the phase which is called as menopause. At menopause or after menopause, the process of oogenesis does not occur. So oogenesis, the process is initiated at or even before birth. That means inside the womb of the mother, the process of oogenesis is initiated, and the first process or the first completion of meiosis, the particular meiotic cycle, it takes years to complete. Okay, years to complete in the sense that the oogonial cells are the one which is starting the process of meiosis, which is called as oogenesis. It starts in the particular female even before birth. That means starting means before, because it is meiosis, prophase 1 will be initiated, and all the cells, that means all the oogonial cells, if you have something around 1 lakh oogonial cells, and in that only most of the cells, they will start the process of meiosis 1 and prophase 1, and these cells will get arrested at prophase 1 of meiosis 1. Okay, arrest in the sense that they will not proceed further in meiosis. They are arrested. In this arrested state, the female, that particular baby, is born. So at birth, if you are observing the ovary, you will have the oogonial cells which have started meiosis, which have entered prophase 1, and there are oogonial cells which are not, which have not yet started the process of meiosis 1. And this particular condition prevails till the particular stage of the lifetime, which is called as menopause, or the first the release of the first oocyte. Till that particular years, or for say if a particular female individual is starting the particular process of menses at the age of 10, what happens is from birth till the age of 10, the ovary is having these two type of cells. That means oogonial cells which have started the process of meiosis, and the other oogonial cells which are still do not have undergone the process. So another important event that occurs in ovary, which does not occur in testes, is which occurs only in ovary, the process which is called as atresia or self-death. That means, as I said, in these particular years after birth till the first menstrual start of the first menstrual cycle, what happens is the oogonial cells which have either started division or without, which have not started division, they will degenerate inside the ovary. They will be self-degenerating. That numbers will be decreasing, and that decrease in number of these particular oogonial cells is usually referred to as atresia. The process of atresia, begins in the ovary before birth. That means when the first meiotic division is initiated, at that particular time atresia is initiated, and that particular event ends at menopause. That means after menopause, there is no oogonial cells inside the ovary. As a result, there is no atresia. It is because of this atresia, all the oogonial cells which are present inside the ovary is getting completely being degenerated, and that results in the process which is called as menopause. Usually occurs at the age of 45s to 50s, and depends upon the individual. And as I said, what happens is, in till 10 years, this particular atresia is happening, and uh, during the first menstrual cycle at the age of 10, what happens is, uh, the oogonial cells which have got a chance to start meiosis before birth, some of them, for say 10 of the oogonial cells, will start the process of meiosis 1. That means they will complete meiosis 1, they will start meiosis 2. Okay, and the second arrest occurs before ovulation, okay, and that particular arrest is at metaphase 2. Okay, at metaphase 2, the arrest occurs. That means reduction division is over. The first the cytokinesis is over. As a result, you get, as you see here, you get two different type of cells. One is a larger one. You might have studied all this, or you might have heard all this before. A larger one, which is the oocyte, and the smaller one, which is the polar body. This is after the first meiotic division. And now this particular oocyte is arrested at metaphase 2 of, not meiosis, metaphase 2 of meiosis 2. And it is at this metaphase 2 arrested state, the particular secondary oocyte or the ova is ovulated. That means without completing oogenesis, the ova is, or the oocyte is getting ovulated. And this particular completion of oogenesis for an oocyte occurs only if it is getting fertilized. Only if it is getting fertilized or during the process of embryo formation, it is completed. Otherwise, in every menstrual cycle, the released oocyte is with an incomplete meiosis. So, completion of meiosis occurs only if that egg is getting fertilized. An unfertilized egg, which is released in every menstrual cycle from the start of menses, if it is at the age of 10, in every monthly cycle of, in every menstrual cycle, the egg which is released is at this arrested metaphase 2 of meiosis. And that means it has not completed the particular phase of meiosis. So that is a major, the main difference between the division of oogenesis and spermatogenesis. Coming to spermatogenesis, as I said, it is a very very fast event. There is no arrest, etc., as we said in oogenesis. At puberty, at the age of 12 or 14, in the case with a boy or a male child, the process is initiated, and that happens at a very rapid speed. That means in every this particular the sperms are produced here. As we said, an incomplete meiosis occurs in the case of oogenesis. Complete process of meiosis happens for every spermatogonial cell. Every spermatogonial cell which is entering meiosis, it completes and sperms are produced. So this is the main difference between the meiosis that is happening between the sperm producing the spermatogenesis as well as oogenesis. So with this session, we are ending the introductory part of genetics. From next session onwards, we'll be discussing the different modules that comes to genetics. So these particular sessions were meant for you to introduce into the topic of genetics, to come across with the terminologies that we'll see, etc. With this, we are concluding our introductory session on genetic terminologies. So in the next session onwards, we'll be directly moving on to the different aspects of genetics. So I hope everything is the ones or the terminologies, etc., that is discussed till here is clear. So we'll come back with the next session, with this the session with the genetic topics. Thank you.